All right, we're up and rolling here. So, first thing I want you to know is that we'll start with prokaryotes. And first bullet point, all living things are either prokaryotic or eukaryotic. There's like this binary classification here of living things. Anybody remember what distinguishes these two? Freshman year, yep. Organelles, yep, that is something. And then how about what are we? Are we eukaryote or prokaryote? We're eukaryotic. Anything that's got organelles, and in fact, anything that's multicellular is eukaryotic. All right, so let's just now that we've established that, okay, let's talk about the characteristics of prokaryotes. And in fact, let's draw one. Let's actually start by drawing one. So draw like a pill-shaped thing. And then label this as the cell wall. And then put in parentheses that it's made of peptidoglycan. Now, why do we care that it's made of peptidoglycan? Because we don't want you to confuse it with the plant cell wall, which would be made of cellulose. This is a different kind of cell wall. Peptidoglycan is a carbohydrate. Okay. The inside the cell wall is a double phospholipid bilayer, right? I don't mean to say double. There's a phospholipid bilayer. And that is called the plasma membrane. So we can put it in parentheses, phospholipid bilayer. Okay. This cell wall has little projections, so draw some spikes. And these are called, this is a pilus. The plural is pili. And these spikes are kind of interesting. So one of the struggles for prokaryotes is they don't reproduce sexually, right? Which means that they have a hard time getting genetic diversity. So what they'll do is they'll kind of, if you can kind of picture one prokaryote bumping into another, and then puncturing the other bacteria with the pilus, like it actually punctures it and injects genetic material. Like they'll actually trade genetic material through that pilus. So this pilus is like a straw. It's like a glass straw, and DNA plasmids are exchanged through the pilus. So I'm not going to draw that. Like I'm not going to draw another bacteria coming over and connecting, but just know that that they would another bacteria could bump into it, and then they can exchange their plasmids with each other. And that's a way that they get a little more genetic diversity, right? Now it's like a kind of a pseudo sexual reproduction, but it's not reproduction, right? It's just an actual like exchange of genetic material. Okay. Also, these prokaryotes, they have this kind of little tail, actually, called a flagellum. And yes, the flagellum is used for locomotion. Not all bacteria have a flagellum, but they can. Another feature is that you're going to find these ribosomes inside the cytoplasm. but they're a specific version. It's called a 60S ribosome, and it is different than ours. Okay, we have a 70S. Another thing you'll see is this spaghetti-like DNA that's just kind of loose and chaotic, but it does form one big loop, and that's called a nucleoid. By the way, anytime you add oid to the end of a word, it means like fake. So it's like a fake nucleus. It's not a real nucleus. Or like remember in our for very first activity, the humanoid, Sophia the humanoid. Oid just means like fake, artificial. Okay, so that's the nucleoid, and let's add that this is naked DNA. The reason we call it naked DNA is because it's not wrapped around proteins. We have a very highly organized way of organizing our DNA, but these bacteria, they just kind of have it all loose. It's just kind of messy. Okay, I think that's it for the drawing. So let's state a few facts. Okay, a prokaryote is either prokaryotes are either bacteria or archaea. Archaea and bacteria are two different domains, and what two different domains of prokaryotes. Archaea. Wow. Let me double check my spelling on archaea. Yeah, it's A E. It's A E. No, E A E. E A E. There you go. E A E. Okay. The other fact that we need to know is they're single-celled only. There is no nucleus. There are no organelles. DNA is naked, which means that it's not associated with proteins. Also, all prokaryotes have one chromosome. And we saw it up here, this spaghetti-looking thing. That's a chromosome. So we can just reference that as the nucleoid. They're kind of referring to the same thing. All right. And they have a... they reproduce asexually through a process called binary fission. They make clones of each other, right? Okay, and then I'm not going to repeat myself, but you can see the pili, the flagellum. So let's talk about the size. They are small compared to eukaryotes. So how small? We're talking 0.1 to 5 micrometers. Okay, so definitely invisible to the naked eye. You can never see a bacteria with your naked eye. Today we're gonna look at bacteria under the microscope. I've got some yogurt over there, and yogurt is primarily lactobacillus. It's a bacteria. We're gonna put it under the microscope, and you'll kind of see these little teeny specks, but it's not going to be all that. You'll really get a sense for how small they are. So that's the size. And by the way, just as a reference, eukaryotes are uh, 10 to 20 times longer across. Like if you measure it across the cell, eukaryote would be like 10 to 20 times longer. And one of the slides today that you're going to try to do is you're going to try to put eukaryote right next to the prokaryote and look at them in the same field of view. You get a little size comparison. So that will be pretty cool. And let's just little sub note, okay? Little sub footnote here. The reason they are small is because they lack intracellular transport and lack compartmentalization. All right, I got a funny analogy here, but when you think of all the business that a cell has to take care of on a given day, right? Getting rid of waste, bringing in food, moving stuff around, processing the food, making the energy, making the what are they called? Proteins and enzymes, and facilitating all of that. It's kind of like a school, right? This school has all kinds of moving parts, right? There's different departments. There's the facilities. There's the maintenance. There's the admin team. They have to be in charge of scheduling events. And then there's the teachers, which kind of do their little jobs. And then there's the students that play a role, right? And all of the above, right? Imagine for a moment what this school, and by the way, the school is the cell. Okay, this whole school is the cell. Imagine for a moment if everybody shared the same responsibilities. In other words, like the administrators, they had to do the same amount of teaching that I do, and I had to do the same amount of administrating as they do, and everyone did everything. Like if everybody just did everything, like in other words, I actually have some facilities responsibilities too. And in other words, there's no specialization, there's no division of labor. It's just like everybody just does everything. It'd be very chaotic. It would be very inefficient. The size of the school would be pretty limited. You wouldn't be able to have a very big school because the inefficiency would limit you. Right? But as soon as you start specializing, okay, you're in charge of that, you're in charge of this, all of a sudden the whole entity becomes more efficient and then can grow, right? You could grow the school and really big if you're as long as you're efficient and everything's getting done, taken care of. So think of this cell up here, this prokaryote, as kind of just this. There's not a lot of specialization. We don't even have any organelles. So all of the responsibilities are all just happening in this soup. There's just picture just this soup right here, and like all the different things are all happening in the same place. There's not even any classrooms. There's no offices. There's no storage rooms, and so I think you get the analogy here. But this is what limits prokaryotes on their size, and we said that there at the bottom. Okay, so now eukaryote. Couple things to say about this. We're not going to draw a picture because eukaryotes have a lot of variability, and there's plant cell, animal cell. Those have differences. So no picture here. We're just going to say a few things. This would be plants, animals, and fungi. Fungi, actually, is how you should say it. By the way, if you look at the etymology of the word here, let's break it down. So "eu" means true, and karyo means like kernel, but they mean it as nucleus. So a eukaryote is a true nucleus. A prokaryote is a fake nucleus. Pro actually means before. So pro means before the nucleus what evolved, right? You had prokaryotes. But now we have true nucleus, and that is the eukaryotes. So yes, they have a nucleus. Yes, they have organelles. Yes, they have multiple supercoiled chromosomes. And we're also going to say that the DNA is wrapped around histones. It's organized around these histones, which are proteins that help organize the DNA. And then it's got, instead of a 60S, it has a 70S ribosome. And can be single-celled or multi-celled. Now today, you'll look at both. You'll have, there's some stuff over there that's multi-celled. There's some stuff that's single-celled. So, should be good. All right, any questions about these? Yep, Grace.
What's the word after DNA wrapped around?
Oh man, I thought you had like some deep question, but it was just, Mr. Roth, can you write better? Raptor, this one?
Yes.
Histones.
Histones. Okay.
Yeah. It's like this little protein that the DNA wraps around. So, yeah, I need to work on my N's, my letter N. Histones. Okay, I'm going to go ahead and stop this.